Today's topic is GenomeStudio genotyping, creating custom cluster files for Infinium arrays. Let's get started. Our goals for today, I'd like to go over with you the basics of clustering in GenomeStudio. What is clustering and how do you carry out clustering in GenomeStudio? We want to talk about when you want to create and use a custom cluster file, why would you want to use it, what situations, and what situations would you not. Also, we want to know how to create and export a custom cluster file from a GenomeStudio project. So these are our goals for today. What is clustering? In order to understand this, you have to understand the Infinium assay. The Infinium assay is used for determining which of two alleles, allele A or allele B, are present for a given SNP in a genomic DNA sample. The assay generates signal intensities for each SNP assay in two channels, signal A and signal B. Genotypes are determined based on the signals returned from the two channels. If you were to look at a SNP result in a Cartesian graph, signal A on the x-axis, signal B on the y-axis, genotype AA would yield signal from all signal A channel and none from signal B. And genotype BB would similarly yield signal from signal B channel only. Heterozygous genotype would yield signal from both channels. If you plotted multiple samples in an assay, you would see that these samples would tend to cluster in the same positions on this plot. Another way to look at this data would be to convert the Cartesian coordinates to a polar coordinate system where theta would be the angle from the x-axis where the data point would be, and R is the normalized intensity, which is the sum of the signals from genotype A and genotype B. I like to think of it a little bit differently. If you look at the x-axis, on the far left you would get signal from A and the far right signal from B. So the x-axis scale would be the total proportion of signal that is due to signal B. So now let's take a look at how these data get clustered and genotypes assigned. There are two algorithms that are largely responsible for this. The clustering algorithm is called GenTrain. It determines the location and shape of each genotype cluster. The other algorithm, the calling algorithm, is called GenCall. Once the clusters have been established, each individual data point is analyzed and assigned to a particular genotype based on that data point's position relative to these clusters. At the beginning of any genotyping project, when you're creating it, there are two options that are presented to you for clustering. You can either import cluster positions from an existing cluster file, or you can ask GenomeStudio's GenTrain algorithm to go ahead and cluster the SNPs based on the data points in the cluster. And our topic of discussion today is figuring out which of these two options you want to use. So what happens when you actually cluster a SNP? I've created a project here in which all of the SNPs in the project have not been clustered at all. So no genotypes have been assigned, no data has been analyzed. The project itself just contains intensity data. So in either the full data table or the SNP table, we can click on the SNP to select it to cluster, and then right-click in the table. In the pop-up menu, we can select Cluster Selected SNP. Now notice what happens after that clustering has been done. There are several changes that have occurred. You can see for this particular SNP that genotypes are now assigned. There are many changes in the SNP graph. And in the samples table, which shows sample statistics, there are several columns which are now red. Let's go through some of these changes. In the SNP graph, GenTrain has now defined the centers of the clusters, and these centers are called centroids. They're denoted by a plus mark in the graph, which is very hard to see at this point. Also, the GenTrain algorithm has drawn ovals around the centroids at 1 standard deviation distance from the centroid in order to define the clusters. Further, call regions have been defined. You can see these darker shaded areas. These are areas within which data points are actually assigned genotypes. So any sample data point that falls in the shaded region is assigned the genotype that is associated with that shaded region, AA, AB, or BB. Some key points about clustering. If you look from SNP to SNP to SNP, the cluster positions are going to vary. Depending on the design and sequence of the probes, those intensity ratios are going to be different. There are also other genetic factors. For example, there may be underlying SNPs in the target area of the probe. There may be other homologous regions which can bind potentially to the probes and cause some effect to the actual cluster positions. On the other hand, for any given single SNP assay, the data clusters, they tend to be consistently in the same locations on the SNP graph from batch to batch. So if you ran another batch of samples and looked at the same SNP assay, the cluster positions would tend to repeat and be in the same places, which is a key feature for these SNPs. Let's move on to the samples table in the project after clustering. Anytime that information in either the SNP table or the full data table has been modified, when SNPs, for example, are clustered or if the clustering has been changed, multiple columns in the samples table will turn red, indicating that the statistics for the table have to be recalculated. If we were to simply update the SNP samples table every time we made a change, it would bog down the process. So we allow the user to make multiple changes to the SNPs and to multiple SNPs before updating the sample statistics. There is a calculator button that appears in the top left, the far left of these buttons, and that's also an indication that things need to be recalculated. Click on the calculator to recalculate the statistics. So in this particular case, we've only updated one SNP. All the other SNPs have not been clustered yet, have not been analyzed. Recalculated statistics are calculated based only on SNP assays which have been clustered. So SNPs which have not been clustered have no effect on the call rate. Now these processes, GenTrain and GenCall, are assigned scores. These scores provide quality metrics for each SNP and for each genotype call. So GenTrain score is SNP specific. It measures how well an individual SNP assay fits the clustering model. In other words, how well does that SNP assay compare to an ideal SNP? The GenCall score is calculated for each individual data point, so it's SNP and sample specific. It measures how well an individual sample data point fits into a given cluster. How close is it to the center of the cluster? It's also a function of the GenTrain score. In other words, the GenTrain score has an influence on what the final GenCall score will be. You can find the GenTrain scores in the SNP table. You can see here for this particular project that there is a column called GenTrain score. And you'll notice in this project that we've created, only the one SNP that we've actually clustered has a score assigned to it. Only SNP assays which have been clustered will have statistics updated. GenCall scores are found in the full data table. The full data table lists each individual sample, it lists the genotypes that have been determined for each sample, and it lists the score along with other sample and assay-specific statistics. So the GenCall scores are found associated with each sample in the full data table under the score subcolumn. So keep in mind it's a GenCall score, but the column itself is simply called score. Again, the GenCall score measures how well a sample fits into a given cluster, and it is a function of the GenTrain score. You can see here, the darker shaded areas are what we call the call regions, and they are displaying the GenCall threshold for calling genotypes. Now, this threshold is by default 0.15 for Infinium assays. Anything that falls within the shaded region has a minimum GenCall score of 0.15. The closer the data point is to the center, the higher the GenCall score. So the high GenCall score samples will lie near the center of the cluster. Any data points that fall outside of that shaded area are data points that have GenCall scores below the threshold. Therefore, they have no genotype and they have no score. Now, you can set the threshold for the GenCall score by going to the Tools menu, selecting Options, and then selecting Project. In the pop-up window that appears, you'll see at the top left that there is a no-call threshold, and by default it's set to 0.15. If you change the threshold, then you will change the size and shape of the call areas, the shaded areas in the SNP graph. Increasing the threshold will shrink the areas. Decreasing the threshold will increase the call areas. If you set the no-call threshold to 0, then the shaded area, the dark shaded area, fills the entire space of the light shaded area, and it means that all data points will have a genotype assigned to them, whether it's proper to do so or not. We strongly recommend that you not set that threshold to 0. You set it to our recommended 0.15 or higher. Now, for clustering basics, let's recap. For a given SNP assay, samples tend to cluster together based on the genotypes. The cluster positions for the SNP assays are generally consistent from batch to batch. Clustering and genotype assignments are carried out by the GenomeStudio algorithms GenTrain, which handles the clustering, and GenCall, which handles the genotype assignment. GenTrain and GenCall processes are scored for each SNP and for each data point, respectively. A GenCall threshold can be set for genotype calling, where samples above the threshold get called, samples below the threshold do not get called. Finally, sample table call rates and other statistics are based only on SNPs which have been clustered. So when should you create and/or apply cluster files? Well, what is a cluster file? A cluster file is a file exported from an existing GenomeStudio project that has been clustered. So the cluster file contains all the cluster positions, also includes the mean and standard deviation of the intensity, and the theta for each genotype cluster in the normalized coordinates for every SNP. It contains the cluster score information, the GenTrain score. And it contains the cluster separation score, which is another statistics that gives you quality. It tells you how separate the cluster ovals and centroids are from each other. The better the separation, the higher the cluster separation score. The cluster file is generally imported into a new or existing GenomeStudio project that is based on the same bead chip type. So if you create a cluster file from a project, you can import it into another GenomeStudio project as long as that project is based on the same type of chip. So it substitutes for the cluster all SNPs step, providing all the information normally generated by the GenTrain algorithm. If you think about it, a GenomeStudio project goes through two steps: the clustering by GenTrain, and then the GenCall by GenCall, the SNP calling. Importing a cluster file allows you to skip the GenTrain algorithm and go straight to GenCall, because we know that clusters are consistent between projects. So if you have a cluster position file, you can skip GenTrain. Illumina has created several standard cluster files for use with their off-the-shelf genotyping products. All of the Omni chips and all of the chips which have been commercialized, such as the global screening array, the global diversity array, and such, Illumina has created standard cluster files based on a large number of samples which we assayed in-house. These have been created using a diverse set of a minimum of 100 samples These are all from different ethnic backgrounds, Caucasian, Asian, and Yoruban HapMap samples. These files are downloadable from the support page for each different chip. You can use the Illumina generated cluster file if the genetic diversity of your sample set is well represented by the standard cluster file. You can use the standard cluster file if call rates and CNV metrics are meeting your expectations after you've applied them to your project. You can also use them if there are a low number of samples in your project. And in general, when should you use a cluster file, whether it's been generated by Illumina or by another scientist in the field who has analyzed the same type of data that you are analyzing? You should use a cluster file when your project has a small sample number. A small number of samples will not provide a statistically significant data set to establish accurate cluster positions for your project. In other words, there's a lot of data missing from a project with a small number of samples. And so it would be best to use a cluster file for a project that has such small sample numbers. If your project is extremely large, for projects with tens of thousands of samples, if you took all tens of thousands of samples in your project and asked GenomeStudio to cluster the data based on the data in this project, it could take potentially an inordinately large amount of time, from several hours to several days, and it may indeed even crash the computer or the program before the clustering is finished. So you'd want to use an existing cluster file for projects which are extremely large. You also may want to use a cluster file when your bead chip contains a large number of rare variants. Just the same way that a small sample number does not give you a statistically significant number of data points to calculate cluster positions, if most of the SNPs you're assaying are rare variants with very low minor allele frequencies, it would take a large number of samples to create a statistically significant representation for those rare variants in order to potentially capture the correct cluster positions for alternate SNP alleles. Chips such as the Infinium exome 24, they have a high percentage of rare variants. We have generated a cluster file for that chip using a large number of samples in order to provide statistically significant data and positions. So other reasons why you might want to create a custom cluster file? Well, for non-standard chips, for custom chips, chips that you've designed and asked us to create, we do not provide cluster files. Custom arrays and consortia arrays are not provided with cluster files. Clustering is left to the customer to do. Alternatively, you may want to create a custom cluster file if an existing cluster file that you've applied does not fit your data well. You may get lower call rates. If you're doing CNV analysis and you're taking a close look at your allele frequencies and intensity data, and the cluster file does not fit correctly, you will see noisier data, and you may not be able to do an accurate copy number variation analysis. For example, differences in your population from the HapMap populations. For example, we've created multi-ethnic arrays that are specific to populations that are not part of the general HapMap, then those cluster positions may be affected by those genetic factors that I spoke of before, underlying SNPs that don't exist in the HapMap, other potential reasons, localized reasons, or population-specific reasons that might affect the cluster positions of your particular data set. Rare variants could also be a reason that you might want to create a custom cluster file, as mentioned before. FFPE samples, the quality of the samples that you load onto the chip will also affect how those clusters are ultimately positioned on your SNP graphs. We suggest that for FFPE samples that you would need to do a custom cluster file and calculate the cluster positions based on FFPE-derived samples similar to what you're analyzing. Some people analyze whole genome amplified samples, which are, again, very different from standard isolated genomic DNA. So those clusterings would have to be analyzed separately and clustered differently. If, for example, your normal variation in processing is different from what Illumina has used in their own laboratories. So every laboratory has potential differences in how things are done. temperature calibrations of the equipment, volume calibrations of the equipment. So you could try to use Illumina's clustering and cluster files, but it might be that there is enough differences in your processing that the cluster positions that we've calculated on our samples may not match yours as well as you'd like. So for all these reasons, you may want to consider creating a custom cluster file. And processing your samples and clustering on your data, what samples should you use? You should use normal samples. You should not analyze tumor samples, because what you're trying to do with clustering is trying to establish the expected cluster positions. And you should establish those expected cluster positions based on normal samples. Abnormal samples are going to cluster differently, and you want to be able to visualize and detect those differences. So you should use normal samples. You want to have samples that are of comparable quality. So you want to make sure that, as much as possible, that the same type of sample isolation, DNA isolation, has been done on the samples, all the samples in your project. You want to use samples that are representative of the population you're interested in. You want to use samples of the type and source that you're going to be studying in order to create your cluster files. And also you want to include as many replicates and, if you have them, parent-child Mendelian analysis samples as possible. This can help to confirm the quality of your data. So in order to capture the most representative data points for your clustering, you want to consider the following. You may want to use a minimum of 3 different runs. You may want to use a minimum of 3 different reagent lots if you have them, because reagent lots may have some effect on your cluster positions. You want to use runs from a representative number of operators. For example, if you have 3 operators in your laboratory, you want to use runs from all 3 of those operators. Differences in technique may account for slight shifts in the clustering. If you have multiple robots that are running the automated portions of the process, you want to use runs from all of your robots. All right? If you don't, if you're only using one of those runs, instead of trying to capture the variations, you can see that the establishment of a cluster position in run 1, and if you run runs 2 and 3 afterwards, you might miss or lose some of the representation. So again, the idea here is to capture normal variation in your laboratories to make a robust cluster file. All right? So how do you execute a clustering in GenomeStudio? Now, I'm going to present some general principles, and the workflow itself is only a suggestion, not intended to represent an absolute recommended procedure for clustering. But we do recommend that you carry out at least these general processes. So in GenomeStudio, when you cluster the project, start with doing a preliminary clustering of all SNPs. And then you're going to evaluate the samples and remove any outliers, any samples that show poor call rates or abnormal performance relative to the majority of the samples in the project. The next step is to actually recluster all of the SNPs, separating out the genders. So you're going to cluster sex chromosomes such that males will be only used to cluster the Y chromosome SNPs and females to cluster the X chromosome SNPs. Both males and females will be used to cluster the autosomes, the non-sex chromosomes. Finally, after doing this second round of clustering, we're going to review, edit all of the SNPs as possible. We'll use filters and scores from different parameters to evaluate the SNPs and the quality of the data from those SNPs. We're going to correct and zero or exclude as many SNPs as needed. So the general process, preliminary clustering, a reclustering where you separate out the genders, and then finally reviewing and editing the SNPs in order to vet and remove or edit as necessary. So let's start with clustering all the SNPs and evaluating the samples. In the GenomeStudio project, at the end stage, right before you are going to execute carry out the project, you have the option, as I mentioned, of either clustering all SNPs or importing an existing cluster file. So in this case, we are going to ask GenTrain to cluster all SNPs and then click on the Finish button. Now, you can, at any time after the project has been created, always call on GenTrain to do it again. If you have clustered the SNPs and then done some editing, but you decided you want to redo that and reset to the beginning, simply click on the Cluster All SNPs button, which is the tricolor button at the top of the page. And GenTrain will then overwrite any previous clustering and reestablish the GenTrain cluster positions. Now again, once you've done the clustering, you'll see that all of the columns in the samples table have turned red. You can click on the calculator to calculate all the call rates. Now, once the calculations are done, you'll have call rates for all the samples. Take that call rate column, highlight it, and click on the Sort A to Z to bring all the low call rate samples to the top of the table. Now, to get a graphic view of the call rates, use the line graph tool, which is the button that looks like two mountains, to visualize the call rates and select the poorest performing samples for exclusion. So let's click on that. What we see here in our project is that there's a couple of data sets here. There's a separation, and this separation here in the middle and the right represents the difference in call rates between male and female samples. This particular chip has a lot of X chromosome, and so when you're genotyping males and females, you see a difference in the call rates. However, this differential is inconsequential to this step. What we're looking for is a low call rate outliers. So you can see here that they are on the far left of the graph. If you click and drag in the graph, you can actually select them in the graph, and simultaneously the samples in the GenomeStudio project will also be selected. Now, once they've been highlighted, right-click in the samples table and select Exclude Selected Samples so that these samples will no longer be calculated in the calculations for statistics. It does not delete the samples, but it removes them from analysis. Excluded samples can be subsequently re-included and recalculated. When asked if you want to update the SNP statistics at this point, answer no, because we're going to be doing a lot of clustering. We don't need to update all the cluster statistics until after we've completed all of our clustering work. So at this step, we've eliminated the samples with poor performance from analysis in the project. The next step would be to cluster the sex chromosomes based on gender, and then cluster the autosomes as well. So after reclustering, after excluding all the poor performing samples and abnormal samples, it leaves only normal high quality samples to establish accurate cluster positions. X chromosome SNPs will be clustered with female samples, Y will be clustered using only males. Autosomal SNPs will be clustered using all samples. So in the SNP table, let's go to the chromosome header and then click on the A to Z button to sort the SNPs by chromosome. So all the chromosome 1 SNPs will appear at the top and chromosome Y SNPs will appear at the end. In the SNP table, click on the first SNP in the table, then scroll down to the last SNP on chromosome 22, and in any mitochondrial SNPs if you want to include them in the clustering, and then shift-click to select all the SNPs except for the SNP sex chromosome SNPs. Now, right-click in the table and select Cluster Selected SNPs. So what we're doing here is clustering the autosomal SNPs. From chromosome 1 to chromosome 22, they've all been selected. We right-click and say Cluster Selected SNPs so that all the chromosomal SNPs between 1 and 22 are clustered. Answer no to updating the SNP statistics for the same reason, because we don't need to update the SNP statistics until after we've done all the clustering that we're going to do. Now, if your project doesn't happen to list the genders for your samples, you can ask GenomeStudio via the algorithm GenderEst to actually determine the genders of your samples. In the samples table, select all the unexcluded samples. So the excluded samples are grayed out. Select all the samples which are not grayed out. Right-click and select Estimate Gender for selected samples. Now, answer yes when you're asked if you want to populate the gender column with the results, only if these genders are as yet undetermined. If your genders were determined, you wouldn't want those genders to be overwritten. What you probably want to do is compare your results from GenderEst to the actual assignments that you originally placed in the project. But in the case that you don't have the genders, answer yes when asked if you want to populate the gender column. So you exclude all the female samples now. Go to the samples table, sort the gender column A to Z to sort the genders, and then select all the female samples and exclude them. Now, cluster the Y chromosome SNPs. In the SNP table, select all the Y chromosomes. They should be easy to isolate because you've sorted them by chromosome. They should be at the very end of the list. Select the Y chromosomes and say cluster selected SNPs. You're using male samples to cluster the Y chromosome. To do the X chromosome SNPs, do exactly the similar thing. We'll first need to re-include the females. So in the samples table, select all of the previously excluded female samples, right-click on the table, and select Include Selected Samples. After you've re-included the females, select all the male samples, then right-click and exclude them. Switch to the SNP table and highlight the X chromosomes, then right-click and select Cluster Selected SNPs. So you are now clustering the female SNPs and the X chromosome SNPs based on female samples. Once you've done this, you can select the male samples and re-include them. Now we're not done yet evaluating all the SNPs, so when it asks you if you want to update all the SNPs, say no again. Now we're moving on to the last part of this, where we review and edit the SNPs to see if we can fine-tune, exclude the poor-performing SNPs, and re-edit the ones that need editing. Now, GenomeStudio provides numerous performance metrics for each locus. The metrics are based on the samples for each locus and provide overall performance information for these loci. Now, editing loci manually should be carefully considered. Now, any changes made should be consistent with principles of population genetics and prevent introduction of subjective bias into the data set. In short, you need to find supporting data for any changes that you make. You can go into dbSNP at the NCBI website, looking up an RSNP to see if the populations that are listed there, see if the minor low frequency distributions are similar to the data that you're observing in your project. Now, filtering is intended to review all the potentially problematic loci by setting discrimination parameters. And let me show you what I mean. There are several metrics, as I mentioned, that are included in the GenomeStudio project data. The most useful for me is the call frequency. If you go to the SNP table, the call frequency will tell you the percentage of samples that yielded genotypes. So what you want to do is look for any SNP assays that have poor call frequencies. We do have a suggested cutoff of less than 97%. Anything less than 97%, if you create a filter for that, you'll be able to highlight SNPs that have less than ideal call frequencies. Errors and PPC errors, as I mentioned before, if you have replicate samples or Mendelian analysis samples, parents and children, you may want to look at SNPs that have Mendelian conflicts or show conflicting genotypes between replicates. Similarly, you can look at all these other statistics. Cluster step is another parameter that I find very useful. As I mentioned before, that tells you how much the ovals that define the clusters overlap. And the lower the score, the worse those ovals are and the more overlapping they are. So you can look for low low-scoring cluster step SNPs. There are lots of other parameters here. I would suggest that you look at these numbers and use this as a reference. We'd be happy to answer any questions that you have regarding any of these particular statistics. And it's up to you to decide where you want to establish these cutoffs. You'll find that, depending on different projects or even different chip types, the cutoffs may be different. So, if you were looking at a chip with rare variants, you wouldn't want to use a low minor allele frequency filter because minor allele frequencies would be low to begin with for this particular type of chip. You would sequentially sort the SNP table by the metric columns and review the SNPs around the suggested cutoff values. And the number of SNPs per loci that the filter catches will depend on how discriminating the filter values are. Now, after you've identified the appropriate cutoff values, you can build a filter with those particular cutoff values to identify any SNPs for review or editing. Some example filter settings are included on this slide. Again, you can review this, and you may find that your mileage may vary depending on your chip. Now, in order to refine the clusters, you can do a couple of things. You can run through your SNPs, and any underperforming SNPs, or SNPs for which you cannot obtain reliable data, you can exclude that particular SNP from the analysis. If you hit the button F2, that will zero the SNP. It will turn the GenTrain score for that SNP to 0, and the data from that particular SNP will not be included in any statistical analysis. Alternatively, you can right-click on the SNP and select Zero Selected SNP from the pop-up menu. There is a button on the toolbar that allows you to do the same thing. If you look at the clusters and see that you don't agree with how GenomeStudio has created the ovals and moved the centroids, you can move those things yourself. You can hover over a particular centroid center, hold the Shift key down, and click and drag with your mouse to move any cluster to a different position. You can hover over any centroid edge, any oval, and use the Shift click and drag to reshape the ovals. And you can also click and drag over a set of data points, right-click on those data points, and assign them to a particular cluster. For example, if you clicked on the farthest left cluster of data points, clicked and dragged over them and highlighted them, then you can right-click and say, assign to genotype AA. So SNPs that have been zeroed or clustered or edited will have a 1 in a column called the edited column in the SNP table. There's a column called edited, and it is by default 0. But if you change the SNP in any way away from what the GenTrain algorithm assigned, or the GenCall algorithm assigned, you will see that 0 in this column change to a 1. SNPs that have been zeroed will also have a GenTrain score of 0. So you'll be able to see in the SNP table which SNPs have been zeroed. Now we come to how to export a custom cluster file that you've created. So you've done all the clustering in your project, and you want to use the cluster positions that you've analyzed, you've vetted, you've edited, and zeroed. You want to use this in the future to apply to future projects. So how do you export these data points? Very simple. Go to File, with your project open, go to File, select Export Cluster Positions, and you have the option of exporting the cluster positions for only a subset of the SNPs in your project, or for all SNPs in your project. Generally, you'll want to export the entire set of SNPs. Browse to the location where you want to save your cluster file. I tend to save my cluster files in the same place I save my manifests because they're often used in conjunction with each other. Then click on Save. Your exported cluster positions are saved as a file with the extension .egt, and these are available to be imported into your next project with this array type. Now, I've listed several resources here in the following slides. There is a technical note called Infinium Genotyping Data Analysis. It goes through some good information explaining how you can cluster projects and what good clusters and bad clusters look like. It explains a lot about what a normalized R and B allele frequencies are. So there's a lot of useful information in that technical note. There are also a large number of online courses and recorded webinars available in the online courses and recorded webinars link that I have listed here. Some particular items of interest: the Infinium Genotyping Data Analysis training shows examples of clustering. GenomeStudio performing CNV analysis using CNV partition is also a training video that goes through how you would do CNV analysis using GenomeStudio. GenomeStudio Advanced Analysis Tools is a recorded webinar. Shows how you can calculate replicate and heritability errors and how to import columns, among other many useful tools. There's also a webinar on the Introduction to Design Studio Microarray Assay Designer for custom and semi-custom genotyping arrays. We have several useful bulletins that are specific to microarray on the bulletin page shown here. And I just wanted to remind you all of the agenda that we started with. We wanted to look at the basics of clustering in GenomeStudio, define what clustering is and how to do it in GenomeStudio. And also we wanted to talk about when to create and use a custom cluster file and how to create the custom cluster file from GenomeStudio.